Welcome everyone to week number 3 in Biology 120 Lab. Let's see what we've got going on. So this past week we covered pH, which is a measure of how acidic or alkaline an aqueous solution is. Buffers, a system of chemicals that regulates pH and keeps it at an even number over a particular span of acid or base addition, and their importance to organisms. All organisms, almost without exception, like to have a neutral pH in their cells. And so if there's acid or base in the area around a cell, cells cannot survive. So that's really important. So this week you're going to learn about cells. And ooh, I love me some cells. I really do. Some of my favorite stuff. Looking through microscopes. Of course, you guys aren't going to have access to microscopes, but we're going to do some other things that have to do with cells. So you will do experiments that have to do with diffusion and osmosis. Diffusion. Diffusion is movement of molecules from areas of high concentration to low concentration until an equilibrium is established. So the molecules just spread out until they're evenly dispersed. That's diffusion. And that is a really simple idea that you know. So for example, if you spray a perfume in a room, you will smell it on the other side of that room later on, because those perfume molecules spread out until they're evenly dispersed. And that is without a fan, without air conditioning or whatever. The molecules just do that. Osmosis is a type of diffusion. Osmosis is movement of water molecules across a semi-permeable membrane in response to this idea of diffusion. So water molecules disperse evenly as they can across a semi-permeable membrane, and that is, in our case, we're simulating a cell membrane. Okay, so we'll see how that works. You're also going to learn about different types of cells. So you're going to learn about prokaryotic cells. Prokaryotic cells are bacterial cells. They mean the same thing. So you can say just bacteria, and you know what kinds of cells those are. They are very simple. They are very small. There are no compartments inside a bacterial cell. So there's no nucleus. There's no mitochondria, chloroplast, endoplasmic reticulum, Golgi apparatuses, etc., etc. There are no little compartments in bacterial cells because they are so small. Did I say that there was no nucleus? That is correct. So the DNA is just all nice and folded up inside the cell, but there's no membrane surrounding it. So we got bacterial cells that are nice and simple and small, and then all the rest of the organisms on the planet are eukaryotic. Eukaryotes is what you call them, and eukaryote means true nucleus. So these cells are bigger, and they do have those compartments, like a nucleus, like several mitochondria, chloroplast, if you do photosynthesis, etc. Okay? So eukaryotes have bigger cells, they're more complex, and they're just different from bacterial cells. Why is this important? Because antibiotics, which kill bacteria, have to have some kind of target in bacterial cells that are not in our cells. I used to like to tell my microbiology students, you know, I can kill any bacteria in you. I can load you up with arsenic, just a ton of arsenic, and I can kill those bacterial cells in you. What's the problem with that? I'm going to kill your cells too and kill you. So antibiotics have to have the effect that they kill bacteria, but not your cells, right? So there have to be different targets for these chemicals to work on bacterial cells and kill them and not harm us. So that's one reason that you have to know the difference between these two types of cells. Just for the pure fact that every one of us has taken an antibiotic before. But there are lots of other reasons too. And then there are viruses. So we covered those in week 1 in lecture, if you're taking lecture. So viruses are just little packets of genetic information. And that's all they are. They are not cells of any type. They're just little packets of genetic information. They get inside cells. They are basically pirates that board mothership cells and take over and cause everything in that cell to do what the virus wants it to do. It's just a packet of information, just like a virus is on your computer, right? A virus on your computer is not another little computer that sneaks in your house and takes over, is it? It's just a little bit of information that comes from the outside through an email message or text message or something, and takes over your computer and makes it do things that you don't want it to do. So viruses are like that too, just little packets of genetic information. And I'm going to talk about COVID and SARS-CoV-2 in just a minute. You're going to learn about the structures in cells and viruses, and you're going to make them. You're going to make a bacterial cell. You're going to make a eukaryotic cell. You're going to make a virus, and then identify it by using the dichotomous keys that are supplied in the lab instructions. What is a dichotomous key? It is a key where you're given two choices. You can do this or you can do that. So if you're just starting out, so is this a large cell with compartments? That would be eukaryotic cells. Is it a small cell without compartments? That would be prokaryotic cells. So depending on which one you choose, then there are going to be choices for prokaryotic cells. Is this cell shaped, has a round shape, or does it have a long shape? Okay, so round-shaped bacterial cells would be coccus, and the longer ones have a couple of other choices, but if it's long, then that's going to be branching out. So dichotomous just means you got two choices. So you're going to use those. They're easy to use. Just follow the directions there. Okay, I wanted to show you this. This is how osmosis works. So take a look at the beaker on the left-hand side, okay? Remember that osmosis is the diffusion of water across a semi-permeable membrane. Okay, the semi-permeable membrane, like your cell membrane, allows water to cross it, but doesn't allow a lot of other things like sugar molecules to cross it. So take a look at the left-hand beaker. You see that you have two sides of the beaker that are separated by a semi-permeable membrane. There are many more water molecules on the left than on the right. There are fewer solute molecules, like sugar, for example, on the left than the right. Okay, so just keep them straight. The water molecules are the blue ones. You got more on the left than you do on the right. All right, after osmosis takes place, look at the right-hand side of your slide. After osmosis has taken place and that water moves, which way is it going to move? It's going to move from areas of high concentration to lower concentration. So from the left side of the left beaker to the right side, and look what happens on the right side. So then you get a difference in amounts in the beaker if you have that semi-permeable membrane, because the water has moved to the right side of the beaker in order to evenly disperse. This is what happens during osmosis, and this is what happens with cells. So let me tell you a funny story while we're here. I had a senior microbiology student, so she was just about to graduate, and she was working with me in my lab at Clemson, where I was doing research, and she was helping me. And so she was counting white blood cells under the microscope because we had gotten a blood sample from someone, and I was doing research on the white blood cells. So her job was to count the white blood cells. She's sitting in a microscope behind me, and she says something. She says, "Janie, there were only 4 white blood cells on the left side, on the left grid that I was counting, and I moved over to the right side. There should be an equal number, and there were no white blood cells. And then I went back to the left, and there were none there. They're all gone." I said, "Heather, did you put the white blood cells in water?" She said, yeah. I said, Heather, remember osmosis? She went, uh-oh. Okay, what happened? So she put white blood cells in water. Where's water in the highest concentration? Outside the cell. You got a cell membrane that is permeable to water. The water moved into those white blood cells and made them swell and swell and swell and swell until what? They burst. Yeah. So that's a practical application for osmosis. And it happens because that's how osmosis works. So you're going to run this experiment with some sugar solutions, and you're going to look at diffusion through your potato experiment. So let me get to that. Before I do, let me mention COVID-19. So what does COVID stand for? The CO stands for corona, because when you look at this virus under an electron microscope, it looks like a crown. VI stands for virus, and D stands for disease. So coronavirus disease, that's what COVID stands for. 19 for 2019 when it was first discovered. Okay. COVID-19 is the disease, the whole process, the respiratory tract infection, and whatever comes along with it. The name of the virus is SARS-CoV-2. You see that down in the description. It is a type of coronavirus, but its specific name is SARS-CoV-2. And what does that stand for? Severe Acute Respiratory Syndrome. Oh yeah, that sounds like what happens. Coronavirus number 2. It's the second one that we have discovered. So SARS-CoV-2 is the name of the virus. COVID-19 is the name of the disease. Here's a picture of the virus, which is really cool. It has an envelope, which means that it picks up part of your cell membrane on the way out of your respiratory tract cells. And that means that it is very susceptible to alcohol. And that's why hand sanitizer works against this particular virus, because it strips off that outer layer of your membrane. It's an RNA virus, positively strand. That just means it acts like messenger RNA in your cells. It directly starts making viral proteins when it gets inside your cells. I know we haven't gotten to that yet in lecture. And it has some spikes that are necessary for attachment to your cells. The vaccines so far have been messenger RNA vaccines, where we put messenger RNA from this virus, at least little segments of it, into your muscle cells in your arm, and those cells start making little parts of the proteins, viral proteins, and then you have an immune reaction to that and make antibodies and T cells and all sorts of things. So that's how the vaccines work. So I just wanted to show you a little bit about SARS-CoV-2 and COVID-19 because it's been in the news so long now, for 2 years now. Okay, so what about the lab? Lab 3. It is going to take you some time. If you look at the time requirements, oh my goodness, more than 3 hours just on the potato and sucrose or sugar labs, and then you got to draw 2 types of cells in a virus. So it is going to take time. So spread this out over the week. The potato experiment is going to take the longest. Even cutting up the potato takes forever. It took me forever. Getting those little tiny pieces. Okay, don't get obsessive about the measurements that they give you. I promise. Because if it says a 1/2 centimeter by a 1/2 centimeter, that's 5 millimeters. That is the teeny tiniest little piece of potato you've ever seen in your life. So don't get obsessive about those measurements, because you will account for that in the table that you enter data into. So the potato experiment is a diffusion lab where iodine diffuses into the little squares and rectangles of potato. Iodine is sort of a light brown color, but when it hits starch, it turns black. Okay, so that's how we're going to know that the iodine diffused into the potato, because normally the potato is white, and all of a sudden we get this black edge all around it, and we want to know how far that iodine diffuses into the potato cubes and rectangles. Okay, so that's that lab. The dialysis tubing with the sucrose solutions is the osmosis lab, because dialysis tubing acts like a cell membrane. One trick about the dialysis tubing: you're going to want to open it up to pour the different solutions in and then close off the ends. You cannot open that dialysis tubing until you get it wet. Okay, so you put it down in the water and then you put it between your fingers like that, and then it will open up. But don't try to open it when it's dry, because you can't do it. All right, things needed for the wet labs that you have this week: a potato, yeah, that would be helpful. Paper, calculator, and a timer. You're going to have all of those. Wear your safety gear, because that iodine is not nice to your skin. I know you put iodine on your skin, but not in this concentration. And just be careful with it, okay? Wear your safety gear, wear your goggles, wear your gloves, all that. You're going to have at least 1 hour of downtime per experiment, because you have to let things work. You have to let diffusion happen, and you have to let osmosis happen before you can measure the results. I give you a hint here, page 24, the last column. Consider what you have to do is outside the tubing compared to the solution inside the tubing, okay? Because that's kind of a weird concept that they're asking you in that part of the lab. Realize that you're putting your raw data in the lab instructions, right? But then you're going to type up all of this information in your lab report. Okay, dichotomous keys, I already covered that. It gives you two choices. You make a decision and you go down a path, right? One thing to remind you about next week, well, you don't know this, but anyway, I'm going to tell you ahead of time that the lab next week has to be started by Thursday, because there's all this prep work that you have to do with the peas before you can run the experiment. You have to soak them, and then you have to keep them wet another day in a baggie, and so that they can germinate is basically the idea. So, you got to give plants time to do the things that they need to do. So realize that next week, lab 4, you have to get it started by Thursday, or else you cannot possibly get it finished. I would suggest you start early in the week for next week and get that going, so that you find out whether you can do it correctly or not, because that one is a little bit tricky. And you need a lot of non-chlorinated water. Just use bottled water. That's perfectly fine, because none of that bottled water has chlorine in it. But don't use water right out of the tap for that, or you will kill those germinating peas and you'll ruin the experiment. Non-chlorinated water. Okay, one thing to remind you: use the lab report forms. I have to grade the lab reports, not the lab instructions. I know that there are tables there and there are questions there in the instructions. Just use that to write down your raw data as you're going, right? You're going to have to be doing so many measurements and stuff, especially with those potatoes. You'll have questions about page number 23 and that table there. If you do, just email me. I'll be glad to explain anything. But I mean, there are 4,000 different columns that you have to fill in, and some of that may not make sense to you. So anyway, put your raw data there, and then in the lab report, then that's where you type in the answers, okay? Include everything in this report. Don't put pictures separately. Everything has to go in the lab report, like the photographs of your drawings, the tables you're going to fill out in Word. So you're going to put all the numbers in those big tables. You're going to see how big the tables are. So include everything in the report, okay? All right, and then you also have to turn in milestone 1 for your final project. Remember, I've mentioned this already. Just look up the instructions and the guidelines for the final project. And milestone 1, you go to Ask a Biologist website. There are 4 suggested experiments, four suggested experiments for you. One that has to do with nerves, air pollution, caterpillars, and ice. pick one that you like, and then you will do an experiment. So 2 of them, the one with the nerves and the one with the ice, you actually do something. The Manduka caterpillars, you look at the data. And the air pollution, you're going to have to look up data on websites to find the data that you need for that experiment. So you're going to have to come up with a research question, put it in the form of an if-then statement, read the instructions for that, and then you're going to be looking at the background information about this particular topic. So what are you looking for having to do with the background information? Think about it this way. If you're doing the caterpillar experiment, why is this important? Why does it matter how long it takes a Manduka caterpillar to pupate and hatch? Why does it matter what the temperature is? Why is it important in the grand scheme of things? You're going to have to think about that, okay? So you're going to answer that question. And what is known about this phenomenon? So what's the normal pupation period for a Manduka caterpillar? Where are they normally found? How long does it usually take them? What's known about what happens to them when the temperatures change? Hint, hint, this may have something to do with climate change. So anyway, just giving you a hint on that one. So why is it important and what is known about this phenomenon? And you're going to write that up for your final project milestone 1, which is due at the end of next week, at the end of week 3. So get started on that, get going on that. You're going to have to look up some background information. Use that Shapiro library because there are a lot of good resources. Don't just Google everything. You know, I've said this before. Googling doesn't always give you accurate information. You have to use reputable sources, so use your library. It's all online. You can find everything, okay? All right. I hope that you have a wonderful week. And as always, let me know if you have any questions whatsoever. I promise you're going to have them this week. m.sigmon@snhu.edu. That's all you need to do. Have a great week.